Namaste, myself Devinshini Saxena, Faculty of Physiotherapy and Diagnostic, and my discipline is Medical Laboratory Technology from JOC Vidyapeeth Women's University, Jaipur. So, welcome in Digital Version 2.0. So, my today topic is bacterial cell structure. So, firstly, they are unrelated to human beings as living things can be, but bacteria are essential to human life and life on planet Earth. So, although they are notorious for their role is causing human diseases, from tooth decay to the black plague, there are beneficial species that are initial to good health. Okay, so bacteria are prokaryotes, lacking well-defined nuclei and membrane-bound organelles, and with chromosomes compound of a single closed DNA circle. They come in many shape and size, from minute sphere, cylinder, and spiral threads to flagellated rods and filamentous chain. They are found particularly everywhere on Earth and live in some of the most unusual and seemingly inhospitable places. So evidence show that bacteria were is extinct as long as 3.5 billion years ago, making them one of the oldest living organisms on the earth. Even old that bacteria are the uh, also can active bacteria, tiny prokaryotic organisms that live only in extreme and deep in the Antarctic ice. Okay, Antarctic ice. Um, and many scientists now believe that the archaea and bacteria developed separately from a common ancestor nearly 4 billion years ago. Millions of years later, the ancestors of today's eukaryotic split from the archaea. In the late 1600s, as Antony van Leeuwenhoek became the first to study bacteria under the microscope during the 19th century. The French scientist Louis Pasteur and German physician Robert Koch demonstrated the role of bacteria as pathogens causing disease. That means the 20th century saw numerous advances in bacteriology, indicating their diversity, ancient legends, and general importance. So, most notably, a number of scientists around the world made contribution to the field of microbial ecology, showing that bacteria were essential to food, web, and for the overall health and earth ecosystem. Okay, so there are two types ways of grouping bacteria. They can be divided into three types based on their response of gaseous oxygen. So aerobic bacteria, they require oxygen for their health and extends and will die without it. And anaerobic bacteria cannot tolerate gaseous oxygen at all and die when exposed to it. And facultative anaerobic prefer oxygen but can live without it. Okay, so the secondary way of grouping them is by how they obtain their energy. Bacteria that have to consume the and break down complex organic compound and heterotrophs. This include species that are found the decaying material as well as those and utilized fermentation and this respiration bacteria can and that create their own energy. So by light and through chemical reaction are autotrophy, so divided of a part of uh, bacterial cells. So show in screen. So wait a minute. So wait. show in this screen. So firstly, capsule. Some species of bacteria have a third protective covering, a capsule made up of polysaccharide. Capsule play a number of roles, but the most important are the keep the bacterium from drying out uh, and to protect it from phagocytes by larger microorganisms. The capsule is a major virulence factor in the major disease causing bacteria such as E. coli and Streptococcus pneumoniae. Non-encapsulated mutant of these organisms are virtual. Example, they don't cause disease. Okay. Second, cell envelope. The cell envelope is made up of two or three layers. The anterior cytoplasmic membrane, the cell wall, and in some species of bacteria, and outer capsule. Okay. And next, cell wall. Each bacterium is enclosed by a rigid cell wall composed of peptidoglycan and protein sugar, that means polysaccharides molecules. The cell wall gives the cell its shape, surrounded the cytoplasmic membrane and protecting it from the environment. It also helps to anchor appendages like the pili and flagella, which originate in the cytoplasm membrane and uh, protrude through the cell wall of the outside. The strength of the cell wall is responsible for keeping the cell from bursting when there are large differences in osmotic pressure between the cytoplasm and environment. Okay, so cell wall composition varies very widely among bacteria and is one of the most important factor is in bacterial species analysis and differentiations. Okay. So next, cytoplasm. The cytoplasm or protoplasm of bacterial cell is where the functions for cell growth, metabolism, and replications are carried out. It is gel-like matrix composed of water, enzyme, nutrient, waste, and gases, and contains cell structures such as ribosomes and chromosomes, plasmids, and the cell envelope encases the cytoplasm and all its components. Like unlike the eukaryotic cells, bacteria do not have a membrane enclosed nucleus. The chromosome, a single continuous standard of DNA, is localized but not contained in a region of the cell called the nucleoids. All the other cellular component and structure throughout the cytoplasm. Okay. Next, cytoplasmic membrane. Okay. So cytoplasmic membrane, a layer of phospholipids and protein called the cytoplasmic membrane, and enclose the interior of the bacterium, regulating called the cytoplasmic membrane, and encloses the interior of the bacterium, regulating the flow of material uh, in and about out of the cell. This is structure pride bacteria share with all other living cells, a barrier that allow Them selectively interact with their environment. Membranes are highly organized and asymmetric, having two sides, each side with a different surface and different functions. Membranes are also dynamic, consulting and adapting to different conditions. Next, flagella. Flagella are hair-like structure and provide a mean of locomotion for those bacteria that have them. They can be found at either or both end of the bacterium of all over its structure. The flagella beat in a propeller-like motion to help the bacterium move toward nutrient away from toxic chemical or in the cases of the phosphosynthetic cyanobacteria toward the light. Next, nucleoids. The nucleoids it is a reason um, sorry a region of cytoplasm where the chromosomal DNA is located. It is not a membrane bound nucleus but simply an area of the cytoplasm where the standard of DNA are found. Most bacteria have a single circular chromosome that is responsible for replication, although a few species do have two or more smaller circular axials. DNA standards called plasmid are also found in the cytoplasm. Next, pili. Many species of bacteria have pili, so small hair-like projection emerging from the outside cell surface. These outgrowth assist the bacteria is attaching to other cells and surface, such as teeth, intestine, and rocks. Without pili, many disease causing bacteria lose their ability to infect because they are unable to attach a host tissue. So specialized pili are used for the conjugation and during which two bacteria exchange for fragment of plasmid DNA. Next, um, ribosomes. So ribosomes are microscopic factories found in all cells, so including bacteria, that translate the genetic code from the molecular language of nucleic acid to that of amino acids, the building block of protein. Protein are the molecular that perform all the functions of cells and living organisms. Bacterial ribosomes are similar to those of eukaryotes, but are smaller and they are slightly different composition and molecular structure. Bacterial ribosomes are never bound of other organelles as they some time are in eukaryotes, but are free standing structure distributed throughout the cytoplasm. There are sufficient difference between bacterial ribosomes and eukaryotic ribosomes that some antibiotic will um, inhabit the functioning of bacterial ribosomes, but not a eukaryotes, thus killing bacteria, but not the eukaryotic organism they are affecting. Okay, so this session is completed, and any query in mention in comment box. So thank you. Namaste.